check I've done that right.
Yeah.
Mm-hmm.
I also test the audio through this, so I'll check it sounds okay. That's probably correct. 17-ish people.
Oh, wow.
It'll just update. We normally have the same, if not more, online.
And can they ask questions through there?
Yeah. People can leave comments, and I monitor the comments in case there's any issues, and I'll ask any questions at the end that anyone says online.
Yep. Great.
Well, good morning, everyone. It's my great pleasure today to introduce Associate Professor Liz Dann. Now, Liz is very well dressed for the occasion, as you can see, and is really promoting avocados. You can even see that on that banner, specially made for this purpose, so which is very good. And so avocados have been on a roll, and Liz just came back from a conference in Colombia, the World Avocado Congress, and she's going to tell us a little bit about what's new in the world of avocados and what role she and the team has played in it. So please, Liz, tell us.
Oh, well, thanks, Andre. And for those who are watching from their computer screens in remote locations, I was cruising the shops at DFO at Jindalee, and JJ's Jeans had this lovely men's shirt, it's a white collared shirt with avocados all over it. So my husband said, you've got to buy that shirt. If you don't buy it, I will. So that's what I'm wearing. So today's presentation is really a bit of a combination of the two talks that I gave at the Congress in Colombia. One of those was on Phytophthora, the other one was on fruit quality. And I've added in a little bit of industry statistics, both Australia perspective and also a little bit on the global perspective. And I've included a few photos at the end of my experiences in Colombia, and also I had 5 days in Mexico after the Congress. So it was a fantastic experience to do that side trip as well. So avocados are they've just gone manic. And I don't think it's anything that we've particularly thought would happen 10 years ago, but they're literally selling themselves, and it's what I'm calling global avomania. The whole world's gone crazy about avocados. So on the left there, we all know the story about the millennials who can't afford to buy houses because they're having too much smashed avo and cappuccino for breakfast. Avocado, this was actually a photo that I took in Sydney. There's this cafe in Sydney called Avocado. People having cappuccinos in avocado skins. I find that hard to believe, quite frankly. And I really like this, especially after just catching a bus across from ESP. Carrot says, let's catch a bus. I avocado. So I thought that was quite cute. Lots of different avocado memes. So a little bit of stats, and this is from the FAO, and those values and numbers do differ a little bit from what our local industry tells us. So from 2017, by far the biggest producer is Mexico. And really, South America dominates the global avocado production. Interestingly, in there is Indonesia, which is 4th ranked as far as global production. This total pie chart of about 20 or 21 countries represents 93% of global avocado production. You can see I've put Australia down here. They're about 20th or 21st in terms of size, and about 1% of global production. New Zealand, even though we hear a lot about New Zealand avocados and we're seeing them in the supermarkets at the moment, they're actually a few rungs lower than Australian in terms of total production, but they export a great deal of their crop, and they do it very well. So apparently, and this is some information that came out of the Congress, volumes sold in the European Union has doubled since 2015. People are really catching on. Retail sales in China are increasing by 72% every year, and I don't know when that actually started from, but people are really going crazy about avocados. This graph was sourced from the Australian, or Avocados Australia Facts at a Glance in 2017, so already it's quite old information. And you can see that the actual production in about 2017 was 65,000 tonnes. The predicted production for 1920 at that time was about 75,000 tonnes. 2 years later, John Tice, who's the CEO of Avocados Australia, said in May this year, already that's gone up to 95,000 tonnes, their prediction. So in the space of 1 year, the predicted output or volume has already increased by 20,000 tonnes. So it's massive. And obviously going into the future, 2024, 25, again here, was just under, 2 years ago was just under 100,000 tonnes. Now it's looking at 115,000 tonnes. And perhaps the industry will need to review that again when they really work out how many trees are in the ground or have a better idea of what production is going to be coming from those trees. At the moment, half the trees in the ground in Australia are under 6 years old. So they're not even bearing to their full capacity yet. That's production. When we look at productivity, that's tonnes per hectare. And in Australia, that really hasn't increased that much. 20 years ago, the average tonnes per hectare was about 7. 10 years after that, it's only increased by a couple of tonnes per hectare to 9 tonnes per hectare. Howard Hall did a study for the Australian industry, a benchmarking study. And in 2015, really the tonnages were still only, or the productivity was still only 9.2 or 9.3 tonnes per hectare. We do know that the best growers in Australia are already producing in excess of 20 plus tonnes per hectare year after year. That's their average. So then the question is, in 2025, can we hope to see an industry average of 20 plus tonnes per hectare? Can we increase it from 9 or 10 tonnes way up to 20 tonnes per hectare? We know that already people in industry are doing that, so the potential is there for that to happen. Why hasn't it happened? And really it comes down to something that Howard summarised and found in his report. The prices paid to growers has gone up 62% in the 3 years that he undertook his study. To me, that means that growers are actually still earning quite good money from having trees that are not producing optimally. And a big factor in that is Phytophthora root rot. And from Howard's report, he and I looked at his database and his spreadsheets and so on. We put a few figures in, and a conservative estimate for Phytophthora root rot and what it costs industry is about $17 million per annum. And that's quite conservative. It's not taking into account what happens to those smaller fruit coming from Phytophthora affected trees. So this was the first talk that I gave at the Congress. There was over 3,000 delegates or something registered. It was quite a large Congress, and it was quite hard to meet up with people as well because of the size of the Congress. But it all ran quite smoothly, and I think most of us were surprised how well it ran given the level of organisation beforehand. So I started the talk in Colombia with this quote. And the preceding info to this is that I was on farm and looking at another issue this grower was having. He said, "Oh, Liz, while you're here, can you just come and have a look at these few trees? I think they've got verticillium wilt." So I said, "Yeah, sure, while I'm here, let's go and have a look." The symptom to me didn't look like verticillium wilt, but I took a few samples and brought them back to the lab and did some isolations. I could not isolate any verticillium from the material that he gave me. So I emailed back saying, no, it's not verticillium, it's probably just early stages of Phytophthora decline. So this was his reply. "Dealing with PC is a never-ending battle. Make sure I'm the first you call when you find a cure." And I like this quote for a couple of reasons. I love it because as a grower, he's recognised that it's a never-ending battle dealing with Phytophthora root rot. You can never give up on the battle against Phytophthora. Secondly, he's optimistic that we will come up with a cure for Phytophthora. So I'm not sure about that, but, but I think it's an integrated approach. So very briefly, Phytophthora cinnamomi is the key organism causing Phytophthora root rot, but we know there are other Phytophtheras and related oomycetes which are also causing the same symptoms and are also just as pathogenic as Phytophthora cinnamomi. And we're just really beginning to get a bit of a handle on what those other species are and their level of pathogenicity. It's not a true fungus, and the key thing about Phytophthora is that its dispersal spore is a zoospore which swims quite readily in free water. And Andrew Manners from DAF has very kindly let me use this little video clip that he took under the microscope. So these are the zoospores swimming around, and these are sporangia, and that one in the centre is releasing more zoospores. So you can see how well they swim in free water. This is just a petri dish of water, and pop, out goes the last one. I like that bit. Let's stop there. So what makes Phytophthora so adaptable to waterlogged situations, high water, over-irrigation, is these zoospores and how they swim. So it's got two little flagella on either end of the spore, and that helps it to swim. They get attracted to root exudates just behind the zone of elongation of the root, and they encyst, which is the lower left photo, and that's where they attack the root from, that zone of elongation. So this schematic comes from a poster which Simon Newitt, a colleague in DAF, is the extension officer based at Marucci Research Station. It's called the Peg Wheel, and that's named after Ken Pegg, who's my mentor and predecessor in avocado pathology world. And so it's very basic principles of integrated management for Phytophthora, and it's named after him. A whole series of things that growers should be mindful of all the time when they're talking about orchard management and dealing with Phytophthora. But it starts before planting. So it starts with good site selection, good site preparation, good rootstock selection, and quality nursery trees. So I put this slide up typically to show growers not to plant too close into boggy, wet areas. Everyone's hungry for space and every little bit of land where they can put these money trees. So down here, it's getting a bit wet down there for Phytophthora and for avocados, and so those trees have died. This was actually on the Comboyne Plateau. It's a water harvesting scheme from several years of drought. They thought it was a good idea to put in this trench to actually retain water. Not a good idea. What does get the green tick is planting up on mounds, and he gets a double tick, this grower, because he's also mulched quite nicely. This central photo, it's a beautiful part of the world in the Comboyne Plateau, inland from Port Macquarie. You can see the lovely hills in the background. When I was away in Colombia, this happened. So it's the same orchard, almost taken from the same position. You can see that range in the background. Massive hailstorms come through and really done a job on this orchard, unfortunately. So we talk about tolerant rootstocks. We don't talk about truly resistant rootstocks because there's no gene-for-gene interaction to enable a true resistance against Phytophthora cinnamomi. And several countries have had very good programs selecting for Phytophthora tolerance over many, many decades. The first one to come out was from the Californian industry, and that was Duke 7. It had a pretty good level of tolerance, and people adapted that very widely. Dusa is the next one, and that's come from the South African selection program. We had both Duke 7 and Dusa in a series of trials, which was completed a few years ago. There's next generation material, which is available now, and a lot of that's come out of the South African industry, but also there's one from our own Australian industry, and that's SHSR04. So this is some data from the last rootstock trial that we undertook, and that was planted in a very high Phytophthora-prone environment. Trees that were old and declined had been removed, so that was a perfect site for Phytophthora evaluation. You can see that on a scale of 0 healthy, 10 dead, Zatano, reed and SS3 are not good rootstocks as far as Phytophthora tolerance goes. The best ones are SHSR04, Deuce is not too bad, and Velvic. Again, survival really drops off with some of these poorer rootstocks. A summary of the work that we've done in Australia that has been published some years ago. Deucer and SHSR04 are highly tolerant. Deucer is available now as a clonal rootstock, and several nurseries are actually selling that to growers. SHSR04 was developed or selected by this fellow here, Tony Wiley, and he has just got that PBR variety, onto PBR. So he has undertaken all the assessments and measurements and so on required to get a rootstock onto under plant breeders' rights, so it's now protected. Velvic is a very good rootstock as well. It's mostly propagated as a seedling. Trouble is that not enough nurseries have access to enough seed. They should have been planting their own Velvic trees many years ago so they'd have plenty of seed. Reed and Zatano are highly susceptible, but there's a lot of seed of reed and Zatano available from both Australia and New Zealand. So in this big rush to plant lots of avocado trees, nurseries are propagating on reed and Zatano. So even though industry's known for a while that they're probably not the best rootstocks as far as Phytophthora goes, that's what they're doing. Bounty is another rootstock which is apparently quite good in high water or waterlogged type situations, but it hasn't been assessed in our conditions at all. So that was sort of pre-planting. Post-planting, there's all sorts of other things that growers need to do to be vigilant for Phytophthora. Soil health and mulch, being careful not to over-irrigate, nutrition, specifically calcium, and no over-fertilising with nitrogen, and also the chemicals. So Ken Pegg always used to say, get everything else right first and then come in with the chemicals. So anything that can encourage root growth and soil health is to be encouraged to growers. Often they won't go and spend money on mulch, oh, we can't get it, oh, it's too expensive, but it really pays off. So this is quite a nice healthy root system. The browning of the roots is just natural root attrition, root decline. Roots aren't white all the time, they do come and go. I did a little study a few years ago where I selected from within one orchard soil from under trees, healthy trees, and soil from under nearby sick trees, and sent that off for microbial estimation, microbial activity. And not surprisingly, I guess, but it was a very consistent trend. The soil from under sick trees had a higher microbial content than the soil from under sick trees. So we don't know what came first. Were the trees sick first or was the soil sick first? I think the two probably go pretty hand in hand. And this is a typical photo of what a tree looks like in serious decline from Phytophthora. So the main chemical in use by industry, and really it was a cure 30 years ago for our industry, is potassium phosphonate and related compounds. It does not directly reduce the Phytophthora populations in the soil. It actually works within the plant. It affects the ability to colonise, the ability of Phytophthora to colonise the plant tissue. We know that it has a dual mode of action, so part of its mode of action is direct fungistasis within the root, but probably a primary mode of action is the activation of plant defence responses. And this hasn't been shown in avocado, but I'm hoping to have a student get awarded an international scholarship to have a look at that. is there plant defences within avocado that are induced by phosphonate, but it's been well known in many other plant species. With phosphonate, you have to time the applications to coincide with periods of maximum root growth because you want the compound to go to the roots when the roots are actively growing. What phosphonate does is that if it's sprayed or injected into the trees, it first moves up in the xylem to the canopy, and then it goes with the photosynthate to the site of the tree which is most actively growing at that time. It's a source-sink relationship, so it goes to the sink. That's why it has to be timed to when roots are growing, because they become a sink. If the fruits are growing and developing, then the fruits become a sink. So this just shows the phenological cycle of avocados, and depending where in the country you are, that might be a little bit different. With root growth, there are basically, this is the brown line, 2 application windows, a short one in the summer, early summer, but then a longer one in autumn going into winter. And this is the primary application window for phosphonate. It's put on as a preventative but also as a curative. So you can bring trees which have declined back to health with a good phosphonate program, as well as your mulching and your careful irrigation and so on and so forth. So because I mentioned that phosphonates can go into fruit, and we know that in summertime that's one of the application windows, but that's when growers do apply phosphonate. In most parts of Australia, fruit are actually developing at that time. So we wanted to know in this current project, what are the phosphonate levels like in fruit? No one had really done a survey of that before. So last year I started a survey and I collected over 40 samples from different blocks around the country. The major production regions being North Queensland, so that's Atherton Tablelands, around Bundaberg, and then southeast Queensland, and then southwest WA represents a quarter or nearly a third of Australian production now. So the levels varied. In Queensland, levels did not get above 100 ppm. In southwest WA, they did climb up to over 200. And we need to really be mindful of this because a lot of WA fruit has received clearance for exporting to Japan, and their maximum residue limit is 150 ppm. So it's just something that the industry needs to be really mindful of as we move to export more of this massive amounts of fruit we're producing out of the country. No problem for fruit domestically consumed and marketed within Australia. I don't think we would ever exceed 500 ppm, I hope not. But certainly for export, we need to keep an eye on things like MRLs for not only phosphonates but other pesticides as well. There's the question, is phosphonate going to lose efficacy because the fungus will overcome it? Really, after 30 years, phosphonates are still very, very valuable, and we have not lost efficacy of phosphonate. There has been some shift in sensitivity amongst populations of isolates. So, for example, I collected a dozen or so isolates from various regions, and they had a different history of high use or zero use or low use of phosphonate, and there's quite a range. But in any population of an organism, you'll get a range in sensitivity to any pesticide. So that's not particularly unusual. So I don't think that we are under threat of losing this compound if it's used properly. And the problems are, they do occur when people misuse them. They say, oh, it's not working anymore, but they probably haven't been putting it on at the right time or at the right concentration. So we're very lucky to have something which is still so effective. Biofumigation is something else we've been looking at in the current project, and that is where brassica cover crops contain a high amount of glucosinolate compounds. When they're macerated and mown up over and over again, and then that releases an enzyme called myrosinase, which breaks down glucosinolates into isothiocyanates. And Tim O'Hare's actually been doing a lot of work on this over several years, and I've been collaborating with John Duff from DAF as well with some field trials. So the isothiocyanates are volatile and they have antifungal and also anti-nematode effects, and they are used in the veggie industry. Potato industry, the strawberry industry actually use these brassica cover crops. So the idea is you grow them up, macerate them in, incorporate them into the top 10 centimetres or so. In our trial, we wanted to look at the effect of putting some black plastic over half the plots to try and trap in that volatile. And here's Emily. Emily ran this trial. She's planting a tree. We had 120 trees to plant after this whole process. So the results of that planting was about a year ago now. And she assessed them on a fairly regular basis. So again, 1 is healthy tree and 6 is a dead tree. The main effect towards the end of the trial, and this was in July, is that the covered plots, the plots which were covered, seem to have healthier trees and better establishment than the plots which were not covered. So this is probably not so much a biofumigation effect, but more a soil solarisation effect, where the propagules of Phytophthora in that top 10 centimetres have been heated up by the plastic, the black plastic, and so deactivated that way. This trial's still on the ground. Now that it's rained, I'll probably go down and assess it one more time and pull up the trial, terminate it, and just isolate from roots to make sure the decline is due to Phytophthora, and then get the final data from that. But it was quite interesting to see that effect. If I reanalyse the data, there may be something with caliente, which is one of the key biofumigant species that veggie industry is using, but it's probably not going to be statistically significant. As far as other approaches or a cure, we've looked at various other things, and my trials are geared towards what can we use now, what's available now, what are other industries using. I get approached by companies saying, oh, can you test this whiz-bang new product against Phytophthora? And I say, yep, okay, and I do that in small consultancy. If it looks promising, then I'm more likely to put it into an industry-funded trial. But my philosophy is if the company isn't prepared to put up a little bit of money to do a little bit of seed work or proof of concept work, then they probably don't have a lot of conviction in what they're doing. There's a lot of new anti-oomycete chemistries around as well, and I've got one of those from Syngenta in my major field trials. So the second talk I gave was on the fruit post-harvest diseases. And again, I've pulled this off Fresh Plaza. There's been a series of avocado stories coming out of the Congress over the last couple of weeks since it concluded. So the Chinese experience is basically consumers are buying fruit, but they're ending up having to throw 50 or 100% away because of quality issues. They're not just disease issues, they are people picking fruit too early, so they're not maturing properly. They've been cold stored too cold, or they've come out of trucks at 35 degrees Celsius, so there's failure other than from post-harvest disease. But you know, he's sort of saying here's this massive, massive market in China, but people are really getting a poor experience half the time that they buy their fruit. So I think we really need to address those sort of concerns from our potential clients. The main diseases causing quality issues are anthracnose and stem end rot, and I'm sure you're familiar with these when you open up a piece of fruit and you see some yucky, stringy, discoloured vascular systems. These two I hope you would never see. Because I'm a pathologist, I like seeing spores and fungus. So this one actually left in the ripening room and forgot about it, and it developed some lovely, the grey fluff is Botryopsferia and the orange one is Anthracnose. Pathologists like seeing that sort of stuff. So I sort of went through the disease cycle in Colombia because there's a lot of new growers in industry. They probably don't realise how these things infect and that they can infect throughout fruit development, but you don't know because you don't see symptoms. So these are spores of Colototricum. They're present on this mummified fruit, which is in the canopy and on twigs and materials that's hanging within the canopy. Splash disperses the spores onto the fruit surface. The fungus germinates and forms an aprosorium and then an infection peg. And at this stage, it remains dormant and resistant to desiccation by high temperatures or anything like that. So it's there. It's already snuck in. And then it's not really until fruit ripen that you see the resumption of growth of the fungus and these disease problems occurring. So you can buy a green piece of fruit, it looks perfect, looks lovely, you don't know it's been infected, but if you let it sit in your fruit bowl too long, bang, that's what happens. All those infections have already occurred in the field, several months before, most likely. So in Australia, we know that there are several different species of Colototricum. And this was a study that Lindy Coates, my DAF colleague, undertook in an ABRIS project a few years ago. So there was 194 isolates in the collection for analysis. They were sequenced. There's at least 9 Colototricum species, but 90% of those were 3 main species, Siamensi, Allianum and Fructicola. And there's a cool climate Colototricum as well. And historically, we've called it acutatum, but there's also Colototricum fiorini. So just because you're growing avos in a cooler climate doesn't mean that you're immune to getting anthracnose. And I think we need to educate some of our growers in the cooler areas about that. We're pulling these cool climate Colototricums out all the time. Stem end rot is caused by many different species. Colototricums can cause it, but also fungi in the Botryosphoraceae, Pestiloteopsis, that's quite a mouthful, and these are its cute little spores down here. I always like seeing those, they look pretty cool. Phomopsis diporthi, and largely they're endophytic infections as well. So they occur within the peduncle of the fruit stem. Again, when the fruits start to ripen, then those fungi resume growth, causing these stem end rot issues. So Eugenie, who's in the audience today, is undertaking a PhD project to look at the range and diversity of these various fungi from all growing areas of Australia. And we're going to get hope to get a handle on which ones are really the ones that are primarily causing stem end rot in fruit, or are they causing the branch cankers, or are they causing branch dieback? And there's several other symptoms that are associated with these fungi within orchards as well. So that's what that she's embarked on doing. Management begins in the field. There's a whole lot of things that growers need to do rather than just rely on post-harvest fungicides or post-harvest practices. So I was asked a question at the session, you know, what percentage do you think actually happens in the field, and what percentage should growers should be putting 100% effort into stopping the disease in the field. But my estimation was that at least 50, probably higher, % of management for this disease should be coming from the field. And really, quality in equals quality out. So if you're putting rubbish fruit into the system, then it's very hard to then try and rely on a post-harvest fungicide to knock out what's there. Better to stop it happening in the first place. As well as the avocado pathogens, there's human pathogens. So at the conference, this was a food safety as well as quality talk. So I did a little bit of a lit search, and there's quite high detections of Listeria, Salmonella, E. coli on avocado skin and pulp. So these were 2 studies done one was done in the US and another from South Africa. So it's important to be mindful of that side of things as well. You know, we've had the rockmelon issue here. It can happen to avos, and what would happen to avos if it happened here to avos? It would just be horrendous if people started dying from one of these horrible infections. No more lovely shirts. Yeah. So I was working with Syngenta, and they actually paid me some consultancy funds to help generate data to get this fungicide registered. So it was a few years of research I did with them. It was registered in 2018, graduate A+. It's a combination of two actives. It's fludeoxonil and azoxystrobin. We found that the combination of the two was more effective than each active alone. And it was particularly effective against stem end rot, whereas the current industry standard for post-harvest is Sportac, prochloraz. There's no claim that it does a very good job against stem end rot pathogens. So there was definitely a need to have a fungicide, post-harvest fungicide, which did the range of both anthracnose as well as the stem end rot pathogens. There are some labelling issues which Syngenta are working through now, because azoxystrobin is also one of our major field fungicides. And there are a lot of fungi which have developed resistance because it's a single site of action within the fungus. It's not happened in Colototricum, and hopefully we never see it happen at all, because it's a very, very valuable fungicide. But we need to be mindful of that anti-resistance strategy as well. So then we ran with Syngenta some trials in grower pack sheds. I'm just going to show one of those. This was a pack shed up in North Queensland. He runs a non-recirculating, which means spray to waste. So the fruit were sprayed in this system for about 30 seconds. Looked at anthracnose and stem end rot. So the Syngenta guy went up there to oversee the trial. Then he sent down the fruit to my lab. I ripened them. Obviously, there's been a difference in anthracnose disease using the graduate, but particularly the stem end rot. Stem end rot in this fruit was really high to start with. Normally, anthracnose is primarily the major disease that we have problems with, but stem end rot was quite high. But as you can see, it's done a pretty good job against stem end rot. Something else that I had a visiting research scientist work with me during last year was electrolysed oxidising water, so EO water. I was quite sceptical about this. I think, oh, well, it's just another one of those things that's a bit of a flash in the pan. But because I'd read something from Roger Stanley, and he used to be here at QAAFI, and I have great faith in him as a scientist, I thought, oh, maybe it's worth having a look at. So this image is actually from a pamphlet that Roger put out. The basic idea is it's analogous to a pool chlorinator, but it separates the analyte and the catholyte. The analyte section is the one that's used for sanitation, and catholyte is a bit more corrosive, and that's used for flushing out pipes and so on for cleaning. It's based on hypochlorous acid, so it is a chlorine-based sanitizer. It's produced from these units on farm or on site. So it's basically a salt, a brine solution, water and power. They're your three ingredients. It's non-corrosive, has low toxicity, and it means that you're not storing large volumes of bleach or quaternary ammoniums on site. So there are some advantages of using it. Once the hypochlorous acid has been used, the solution decomposes back into dilute saline. So that's less of a problem for disposal. It's been shown to have strong antimicrobial effects at fairly low concentrations, and it is already used in hospitals and fresh cut processing. Gourmet gardens are using it. Rugby farms apparently have one of these units, and they're using it as well. So we wanted, this is Kamral, he's a Bangladeshi professor now. Lindy Coates and I co-supervised him for his PhD, which he did at Gatton many years ago. So he came back and did a 6-month Endeavour Fellowship with me. He looked at 20% solutions of electrolysed water. They reduced, in dipping trials, they reduced the severity of stem end rot of avocado, but not anthracnose. So we're not sure why, in that instance, the anthracnose was not reduced. But then the fruit that we sourced for this trial came from an orchard which uses no sprays whatsoever. So the levels of natural infection are really, really high. So you cannot get more diseased fruit than the fruit that we used in this trial. So it was a pretty harsh test. He showed that it was more effective than bleach in the dip treatments. There's no difference in the ripening characteristics of the fruit. EO water is an approved input for organic farming. So when I sort of put this up at the Congress, there's quite a lot of people that came up to me and said, well, we can't use fungicides, maybe we can use this. And although it wasn't tested in his study, because it's a sanitizer, it's likely to have some really beneficial effects against the human pathogens as well. So I'm just going to put up the final trial, the results of the final trial. Maroochy Research Station. So DAF have a station at Maroochy, it's near Nambour. They have a very nice experimental scale packing line. Used to live under the building in the old entomology building, but when we got moved out of that site, it got moved to Maroochy, but it's still operational, which is fantastic. So this sort of simulates commercial practice. There's brushes and there's overhead sprays. So he found that the 20% EO water for 30 seconds significantly reduced both anthracnose and stem end rot, and that in fact, at the 30-second time of exposure, it was similar to the fungicide, Graduate A+. So while the dipping treatment had no effect on anthracnose, probably the action with the rollers may have actually helped the sanitizer get into those infections. I don't really know, and we didn't have really time to follow that up any further. He was only here for 6 months. So really for fruit quality, I think we've obviously got to look at combinations of these types of things for optimal efficacy. Potential for organic systems for EO water alone, but I think we need to do a bit more work on that, or as a pre-rinse, or followed by your Graduate A+ fungicides. Jim Adescavage at UC Riverside, I've been having a bit of dialogue with him lately, and he's looking at other promising treatments. So he's looking at things like natamycin, which is already used as a biopesticide. There's no resistance reported, and he's getting some good results with using that compound as a post-harvest treatment. Interestingly, it's also known as pimaracin, and that's something we put into our media for, our selective media for Phytophthora. So it knocks out all sorts of other things but Phytophthora, so that's a key ingredient with that. He's also looking at various food-grade organic acids. So I think we need to be a little bit smarter if these countries are going to demand, and the EU particularly is going to demand fewer pesticide inputs. We need to look at other things which are not strictly hard fungicides. I finished this by saying the quality and safety of fresh or processed avocados is the responsibility of everyone in the supply chain. So it's not just the post-harvest, it's not just the people selling it at Woolworths, it's everybody, it's the growers, they have a prime responsibility, it's the pack sheds, it's the transporters, everyone really has to play their part to get quality product to market. And given where avocados are grown, we're really stressing that fruit. It's not, in Australia at least, it's not grown under the optimal growing conditions that I saw in Mexico and Colombia. So there's high infections, there's stressed fruit from drought and flooding and hail and whatnot. So we're really going to have to be on top of our game to get good quality fruit to market. So last part of the talk, I'm just going to put a few slides up about Colombia. It's always great to go to another country and see how they do things and get some of those experiences. So the day before the conference started, I took a field tour that was arranged by the conference organisers. This is a chrysanthemum packing facility, about an hour's drive east of Medellin, where the conference was held. So some of our guys thought they'd be smart and they took a photo of this and sent it to their wives and partners and said, "Oh, I thought I'd send you some flowers." So that was very cheeky. This was the only orchard I saw in Colombia. Close spacing, so 3.3 metres. And look, disease. So given the close spacing, disease pressure is there, they're surely going to have some problems with fruit quality and post-harvest disease. They're a very young industry in Colombia, despite having about the third highest production. They're going gangbusters. They are wanting to take over the world as far as avocado production goes. So they're learning from as many people as they can, which is why they wanted to host the conference, to get best knowledge from around the world to enable them to go ahead in leaps and bounds. And if they take on some of the things that we heard at the Congress, then they're probably in a pretty good position to do that. But they do have high rainfall, they have good soils, but they've got disease too. So something which was interesting, it's sort of highlighted to me that there's a lot of insects and bugs and things that these other countries have which may not be on our biosecurity pest list, and I've bumped this on to Andrew Gearing, who has the biosecurity project. This is something called monolonian. Again, Andrew Manners had a quick look on the relevant sites for me. There's no apparent mention of this in Australia. Maybe it's called something else, maybe it is here and we haven't looked in the right places, but it could be quite devastating if we got it in Australia, so that's something we need to be aware of. It's a mirrored, that's what it looks like. In Colombia, they don't have really a rootstock. What they do, and they do this in Mexico too, they go out into the native bush and they pick fruit from the native avocado trees, and that's the seed for their rootstock, which is very low tech. They dip the fruit in thiram, which is why it's pink, and the grafting is very much like how we would do it, and then they spray with carbendazim, and carbendazim's been withdrawn here for several years, so they're clearly not up on their PPE. This is John Tice, he's the CEO of Avocados Australia. So it's always good when the bosses get out there and see what's happening in other parts of the world as well. Mexico was lovely. That was the last 5 days of my trip. It's a very old city. These buildings in the old part are UNESCO heritage listed. This was where I was staying, but you know, the rooms were lovely and flash and inside was amazing, but it was still just $85 a night or something, so it was lovely. This is a viaduct that the city had built to get the water from the mountains back into the city. Lovely old churches. Every 300 metres there's a church. So this is my collaborator and my host while I was in Mexico, Salvador Ochoa. So he's a plant pathologist and he arranged 3 really, really great days for me. This is up in the hills a bit, and this is about 2,200 metres, so a bit higher than what we're used to. This was a town that a volcano had erupted nearby and come through. It wiped out the whole town. Obviously those people moved, except parts of the church. So there was 2 ends of this church that were still standing, and otherwise the volcano, this is in 1945, had come through and wiped it out. So there's big lava flow there. This is in Uruapan, which is where I was staying, which is close to the main avocado production in Mexico. And they'd made a lovely botanic garden and national park out of the natural water flow. So this is actually the water source for the city. And they've just done it so nicely with the little bridges and the paved pathways and so on. And apparently when it gets quite hot there, people just flood to the park, and it's cool and calming. So that was a lovely spot to visit as well. So in Mexico, they're very low tech as well. And I saw some orchards which had nutrient deficiency, they had lots of mite damage on them, and you know, their productivity is not high either. It's probably less than 10 tonnes per hectare. But they produce, you saw on the pie chart, a third of the world's avocados. So if their productivity ever bumped up a bit, then they'd be producing a whole lot more fruit. So very volcanic. This photo probably doesn't do justice to actually how steep this little plug was, and we actually climbed right to the top of that. One of the things I was really keen to see in Mexico was armillaria disease. It's something that as a pathologist in undergrad you hear about, you learn about it, and it's still a bit of an enigma because you never really get to see it. So this tree had died because of armillaria, and this is what pathologists do when they're together. They look at cultures. We do that at ESP. It happens everywhere. This is armillaria. So it's a basidiomycete fungus, and typically, and the literature says, that the one that's infecting avocados forms this clump of mushrooms, the fruiting bodies at the base of the trunk. Doesn't happen with this one because this one isn't the typical one. This one is Gallica, Armillaria gallica, which again I don't think is on our biosecurity list. He's also pulled out Armillaria mexicana, and there's an undescribed Armillaria as well that's causing big problems in avocado. So these stringy sort of structures here are what they call the shoestring structure. They're rhizomorphs. So they are fungal mycelium all bunched together, and they grow through the soil. So that's one way that the fungus spreads without needing a host. It grows through the soil, which makes this pathogen really, really, really difficult to manage. This is a cutting, so you can see one of those roadways going up that steep hill. This is where you can see the rhizomorphs actually growing out of the cutting. Very hard to manage. Again, something which was interesting, we're at another orchard. This was a much better managed orchard. It was managed by a female. Didn't get to meet her, unfortunately. And here's Salvador. I gave him an Avocados Australia cat, which he was very happy with. I said, oh, what's this bulbous thing, the base of this tree? I'd never seen that before. This is a close-up of it. And he said, oh, that's agrobacterium, which then I had a light bulb moment because last year a grower from Western Australia sent me this photo, and then she sent me what she cut off, disguised as a cute little bee. And we isolated a bacterium from it, but I hadn't had a chance to do any sequencing on it. So more than likely, this is the aerial form, and after a conversation with him, he said, yes, it can. It's very rare, but it can form these galls on aerial parts, on branches. So that kind of confirmed to me, yes, it probably is agrobacterium. I don't think it's going to be a threat to our industry or anything, but it's nice to have a bit of confirmation that what I suspected was true. They call this elephant foot for obvious reasons. So that was really good to see that in the field and have that discussion with him on site as we saw it. So he'll be a key contact going forward. We got on very well, and we've already had some email dialogue. So really that's it. Thank you very much for listening. Obviously, a few key people in industry have really helped me out over the years. That's Ken, Tony and Graham Thomas. Cameron had an Endeavour fellowship which allowed him to do the electrolysed water study. We can't do our research without grower collaborators, so it's been fantastic having an industry which is so supportive of research, even if we let their trees get a little bit sick. Several people have helped with the EO water, and again, the Syngenta people and Pack Sheds have helped with the graduate A+ evaluation. So thanks very much.
So are there any questions for this?
Back to Phytophthora, do you either inject or spray? Do you recommend doing both at the same time or at different seasons for one and the other?
So for spraying,
You didn't mention Vitamil either, which I had been using.
Ridamil's great and it's expensive. So Ridamil is very good if you know you're going into a Phytophthora infested area and you're replanting into there. So with small trees, you can afford to put on some Ridamil twice during that early establishment phase. But Ridamil to treat big trees is not really cost effective. Phosphonate, if your trees have a fairly decent canopy and they're reasonably healthy, then yes, you can spray because you need the canopy to drive the translocation of phosphonate down to the roots. Injection, you can use both as a preventative and as a curative. And usually one injection a year should be sufficient. Yeah, and that's in that autumn, winter period. Yeah, autumn, winter is probably best. Yeah. We can talk about that. Yeah.
I started fascinated comparing my experience with animal industries with the avocado industry. So in the industries I'm familiar with, there's been enormous pressures coming from marketing and the supply chain end of it to drive organic, chemical-free, antibiotic-free, hormone-free pressures. So are those things happening?
Definitely, yeah. Yeah. Specifically for the EU, but also for other markets as well. And, you know, New Zealand export a lot, and they can't use fungicides in the pack sheds that they pack avocados from because they also pack kiwi fruit. Kiwi fruit having an edible peel, although it's not very nice, there's a lot of fungicides they can't use post-harvest. So because the same sheds are doing both avo and kiwi, then the avos don't get the post-harvest. So there's a big problem with MRLs, and often the European supermarkets cut again. So the EU might have an MRL, a maximum residue limit, but the actual supermarkets reduce that by half or a third, which it's a big problem. And I think, you know, it's why we need to, I don't think we can change the mindset of Europe or the regulatory authorities, but we do need to get smarter about in-field and also some safer, softer options as well. They probably won't be as effective as the chemistries.
Any other questions?
Just another thought on Phytophthora, sorry, that's my bugbear. Is it in all soils throughout, or different soils have?
We assume that if it's not there, so if you're going into a clean planting area, it will be at some stage. So you should just assume it's there. But, you know, we are getting a bit more of a handle on which species might be present. It's mostly Cinnamomi, but we haven't really looked widely till now. It's worldwide, yeah, everywhere.
Yeah, the effective use of compost in the system, in the whole system, what you would consider to be a good compost, and what ratio throughout the industry do you think is using compost effectively?
I took that slide out because it is more of a management one. Compost's very good. I think a compost has to have a woody matter component. It has to have an animal manure component and has to have a calcium, so a gypsum or a dolomite. And that's, I believe, I don't have a direct experience with testing these different sorts out, but I think that's what's worked most effectively. As far as people actually adopting good compost, might be 10% of industry might be doing it. There are more that mulch, and I think there's probably more that want to mulch if they can get their hands on something suitable. A lot of people are using straw because it's cheap and available, but I don't think that's particularly useful for long-term mulching because it has to be topped up. And I think it creates a bit of an anaerobic layer as it compacts rather than being a loose and open like a woody mulch would be. So I think we need to drive that home because soil health, soil carbon is driven by good compost, good mulching. Bang, that's probably half the battle won.
You were talking about sort of, you know, started off with 9 tons, sort of the average, and then you can get 20 tons. But what is actually the theoretical maximum you can get if you do everything right?
That's a good question. Yeah, and I don't think anyone's come up with an answer like that because it would depend on using the best rootstock for that particular patch of ground, the best growing conditions, so irrespective of things you can't control, so good mulching, good use of chemistries where available, good scion. Probably, I know that some people can get yields of 30, 35 tonnes. I don't think they could do that every year, and I think there's probably a maximum life to an orchard as well, if you're pushing trees so hard to do that.
But there's still then a long way to go. I mean, like, so why is it so low, the yield so low? Are people getting too much for their avocado?
That's what I'm suggesting. Yeah. Well, I think with competition, you know, us having to look to export markets, but there's already Chile in those export markets, there's already Mexico, there's already Peru in those markets. Our product has to be really good and really competitive, and is it going to be?
I'll ask one more, please. Pollination must be so important in any fruit. Is there a sort of weather condition? Because some of my trees have bearing a lot of fruit, and some will have one or two on, which it must be, I think, pollination.
There's a lot of variation amongst trees within an orchard, especially if they're on a seedling rootstock. So each tree can be very different, but also. Well, pollination, there's been a lot of work in industry on pollination. So if you receive talking avocados, there's usually a write-up on pollination in that every issue. So flies, it's coming out that flies are very good pollinators as well as bees. Yeah, so it's quite interesting. Yeah, no, all those things, and there's things underway to try and increase the pollinators in orchards. So not only the bees, but also these native pollinators as well. Yeah, definitely.